Thank you very much. I'd like to first thank the BGRI for the opportunity to talk today and update you on our research program. I've changed the title slightly to molecular diagnostic and surveillance. And so we've been working on this for a number of years. And so this is what I call phase 2. So as Moen's mentioned, our favorite pathogen is Puccinia graminis form specialis tritici, the pathogen for wheat stem rust. I will fondly refer to as PGT during this talk. So when we started out doing molecular diagnostics and using molecular tools to do surveillance, the primary objective was to find out and identify UG99 in the race group and find out where it is and where it's moving to. And there were really only 2 other race groups within Northeast Africa. And we developed a series of what we called the UG99 race group stage 1 and stage 2 assays. And then things began to change. And so in Ethiopia, we had the epidemic of the TK-TTF race in 2013. And then also in that year, we had the unusual event of stem rust in Germany. And we had 6 more players. And also that year and the following year, we had an event in Georgia. And we had more than just 2 or 3 players. And so the picture now has become quite more complex. And so from a molecular diagnostics and surveillance standpoint, we're not only interested in asking, is it UG99? Now we have a whole cast of characters that we want to find out and be able to track. So the first thing to do in terms of developing a new molecular diagnostic assay was to, first of all, genotype. And so what we did was to take representative isolates from these different races corresponding to the selected regions, and we genotyped them on a SNP chip. And this gave us a phylogenetic tree, and these isolates from Georgia and Germany and Ethiopia broke out primarily in what we call clade 4 and a little bit clade 5 and clade 3b. So our task was to develop a diagnostic assay that would be able to distinguish between these different characters and also develop a diagnostic assay that is quick and so it's quick and rapid in the lab, but it's also a technology that's transportable so that it can be transported and used in other regional laboratories. And so those were the objectives when we were going forward to developing the assay. So what we did, from the SNP chip, we defined MLG groups and their corresponding phylogenetic clades. And this gives a representation. So if we concentrate on this cast of characters that really Africa saw in Germany, a little bit in Georgia, we kind of ignored the 2013 clan. We have 11 different MLGs. Now, what I want to point out is that in this case, we have 3 different MLGs. So 3 different genetic groups that all have the same race, that they're all TK-TTF. Okay? So using the SNP chip data, we mine that data to develop a number of candidate SNP markers, and then we tested those initially on 15 PGT isolates. And we came up with a final set of 17 SNP markers that is able to distinguish between each of the different 11 MLGs, in addition to distinguish between known other genetic groups within our phylogenetic tree. So the next part of this talk, I want to show you how we've applied this assay to samples over the last several years out of concentrating on Northeast Africa. So in this program that we developed with the UG99, we developed what we call D samples. And so this is a way of collecting samples, field samples, a single pustule from a stem, preferably, or a leaf, that is then killed in ethanol and then is mailed to us or any other diagnostic lab. The advantage of this is that you don't have to worry about moving live cultures. The disadvantage is you don't have live cultures, okay? But it allows a program to be able to go in more depth and cover more area in terms of samples than if you were just looking at live samples. So first of all, I want to talk about Ethiopia. And so we have 4 years of data, 2014 through 2017. And so I'm going to go successive years. So just so people in the back, if you see the orange block, that means that frequency of that particular clade is 20% or more. So in 2014, we really saw two dominant genotypes. We saw clade 1, which is our friend UG99, and we saw the clade 4b, which is the new Ethiopian TK-TTF. In 15 and 16, we saw a progression where clade 4b took over the population, so it now represented 90% of the population. And clade 1 was not even seen at all by 2016. 2017, we see a beginning of a shift again. So we still see clade 4b, our friend TK-TTF and relatives, as the most dominant, but we see again an increase of diversity. Okay. Now if we move next door, go north to Eritrea and ask, what does that look like? So we have 2 years of good data. And in this case, so we have 2016 and 2017. And again, look at the colored block. And notice, you probably cannot read, but the position, but this is clade 3b. Okay. This corresponds to the race TTRTF. So we don't see clade 4b or very little clade 1. If we go to Kenya and look at the same type of thing, we see a different pattern. So in this case, in Kenya, our friend UG99, clade 1, is dominant every single year, 2014 through 2017. In 2015, you can see the incursion of clade 4b or TK-TTF, and it represents 22%. But it then decays. The next year it's 15%, and the following year is 7%. And if we move a little further south to Tanzania, we see something more similar to what we saw in Kenya, where clade 1 is clearly predominant. But by 2017, it's 45%, and we have 45% clade 4b. So Tanzania in 2017 looks like Ethiopia in 2014. So the question here is, what's going to happen in Tanzania? Is clade 4b going to take over like it did in Ethiopia? Or is something else going to happen? So I want to move on and give a little bit of a blown up geographical perspective for two of the characters we just talked about. So the first one is clade 3b. So this one has this race, TTRTF, was first observed in Georgia in 2014. In D samples, we picked it up in Eritrea in 2014. It also was picked up in Azerbaijan in 2015, Egypt in 2016, Iraq in 2016, Italy in 2016, Ethiopia in 2017, and Tanzania in 2017. So this race appears to be on the move. What it will do in the future, whether it will be the new UG99, the new 4B, and take over populations that we've seen that pattern in the last few decades or not, we will have to see. But it clearly is spread across a fairly wide region. The next one I want to talk about is our friend 4B. And so originally the race was TK-TTF. This lineage, MLG, also now includes the TT-TTF. And as I mentioned, it was first picked up in Ethiopia in 2013. Also that year, it was picked up in the UK. It was also picked up in Azerbaijan in 2013, Iraq 2014, Yemen 2014, and you can see that it has spread quite a bit. So again, the question is, where is this guy going to go? And is it going to be like UG99, which has a limited spread? I mean, that's one of the big questions with UG99 clade, is it really hasn't gone as predicted. It has been relatively restricted to Northeast Africa. This data suggests that these two clades have a lot larger geographical spread and population, and therefore has a more higher chance of significant impact on wheat production. So I just want to make a quick summary. First of all, we've developed a PGT core assay that is quick, it's portable, so it can be used in regional diagnostic labs. We've demonstrated the usefulness of this assay for surveillance for PGT in Ethiopia, Eritrea, Kenya, and Tanzania over several years. The intriguing thing that came out of this is that the PGT population in Northeast Africa is subdivided into regions. And what's happening in Eritrea, and what's happening in Ethiopia, and what's happening in Kenya are all different. They are overlapping populations, and you see movement of genotypes and races between those groups, but they are acting and behaving very, very differently. And the question is why? The next, and so then the other major point here, as has been said many times by very famous people, PGT is a shifty enemy. And we are in the midst of a time in which we are seeing population shifts occurring at relatively rapid frequencies, something that hasn't been observed in the last 50 years. And it's not only in PGT, the same thing is happening in Striaformis also. And so one of the questions that really needs to be addressed is what has changed? Why are we seeing these types of population shifts, rapid population shifts now that we didn't see 20 years ago? Okay. Anybody have the answer to that one? I'll give them a free bottle of water. So, of course, thanks goes to lots of people. Any type of program like this is highly dependent on the wonderful collaborators through BGR Global Surveillance who provide the samples and allow things for this to happen. And of course, people like Eugen and Pablo at CDL and Moens at the GRRC who provide isolates that we can genotype and make linkages. Funding from BGRI and the USDA. And then of course, my folks in my lab who actually do all the work. So thank you very much.